All right, so part one, the first thing that we need to cover is some terminology. So again, terminology is very important. You have to start to memorize these words. And some of the important words describe how an individual looks. So character, trait, and phenotype are all different ways to describe how an individual looks. So character is a single visible feature of an organism. A trait is one possible variant of a given character, and a phenotype is the composite of an organism's observable characters and traits that are under study. So let's describe this. So Mendel was interested in studying pea plants, and there was a bunch of different characters that he was interested in looking at. So seed shape, seed color, pod shape, pod color, flower position, and stem height were the 7 characters that he was looking at. So a character does not specify the variant it takes. It just says that this is kind of the general feature that we can look at. So seed shape is the shape of the seed. And so there are 2 different traits that this particular character can have, either round or wrinkled. Seed color was a second character, and they could have 2 different traits. So the variants of that character were either yellow or green. Mendel also looked at the pod shape. So these are the pods that carried the seeds, and they could be either full or constricted. The pods could also be different colors, green or yellow. The flowers that eventually became the seeds, those could be 2 colors, violet or white. And their position on the plant could be different. So they could either be axial or terminal, so either at the end of the plant or throughout the plant. And then the stem height, the height of the plant, could either be tall or dwarf. So again, characters describe what you're looking at. Traits describe the variant that you're looking at. And then finally, phenotype. Phenotype is the composite of an organism's observable characters and traits. And so that would typically mean you're specifying 2 different features. So you might say this individual pea plant has yellow, wrinkled seeds. So if I did that, I'd be specifying multiple characters and multiple traits, and then the composite of that together is the phenotype. So the phenotype of the organism is yellow and wrinkled. And I could do that for all 7 of those if I wanted to. I can specify the phenotype of this plant is round seeds that are green, full pods that are yellow, axial flowers that are white, and it was a tall plant. So in combination, all 7 are the phenotype. So what did Mendel do? So Mendel was able to control the breeding of different plants, and through this analysis, he was able to understand rules by how genetics works. And so artificial population, so this also stimulates the use of this term, artificial populations. So in some situations, we control the breeding of individuals, and other situations we can't control the breeding of individuals. So if we can control the breeding, then we refer to these populations as artificial population. So the experimenter determines how an individual's mate. So the first generation of this artificial population is often called the P or the P0, and these are the parental individuals that start the cross. Very often, you need to know whether or not these individuals breed true. So if they breed true, then that means that they're homozygous. Parents can differ by 0 or more traits. And so typically, you might start with an individual with a phenotype that is round and green and cross it to an individual that is yellow and wrinkled. The first generation of progeny are called F1. So if we have 2 individuals, and they produce, 4 other individuals. We start with the P generation, and then we go to the F1 generation. And then these individuals can also be used to create crosses. And so if we make more individuals, these are called F2s. And so on and so forth. We can continue to produce additional generations of progeny, increasing the number each time to refer to it. So F3s, F4s, F5s, etc. And so initially, the types of crosses that were performed were simply monohybrid crosses. So monohybrid crosses reveal how one trait is transmitted, and a monohybrid cross is between two true breeding parents. So these parents are homozygous. We'll get to that more in a second. And the parents have different traits for only one character. So by specifying it's a monohybrid cross, what we're saying is that only one character is different between them. And so there's a large number of different ways that we can do that. So if we have an individual that is this particular set of traits, so round yellow seeds, full green pods, purple axial flowers, that is tall, we can do a monohybrid cross by an individual that differs just at one of those. And so, how do we do that? So if we cross it with an individual that has these particular sets of traits, the only traits that are different is that one has round seeds and the other one has wrinkled. We could also do a situation where they only differ at this one right here. So a monohybrid cross, out of all these 7 different characters, only one of those characters is different. And so these allow us to look at just a single set of genetic differences. So this is what he did. So these are the 7 different monohybrid crosses that were performed by Mendel. So in one case, he crossed round seeds with wrinkled seeds. And again, all those others were the same. In another case, he crossed yellow with green, full with constricted, etc., etc. And so in the first case, one of the things that he kept track of is he looked at all of the F1 individuals. So one of the things he found, which was a surprise, was that the F1 individuals all had one of the parental phenotypes. So in this case, the F1s all had this parent right here, and so forth and so forth. So the way we've listed this in the table is we've always listed the trait that the F1 progeny had as the first. And so this doesn't match the blended mode of inheritance. If inheritance was blended, then what we would expect to see is a mixture between green and yellow colors, for example, for these F1 individuals, or a slightly wrinkled seed as opposed to a fully round seed or a fully wrinkled seed. So then Mendel continued to do this cross, and so he crossed the F1 individuals together, and then he found that he could get a large number of different phenotypes in the F2 individuals. So the F2s were no longer all identical to each other, but in fact some of them shared the first parent and some of them shared the phenotype of the second parent. So again, he never saw a blended mode of inheritance. All of these individuals had one of the 2 parental traits. Further, he found a very specific ratio, and this ratio was very close to 3-to-1. So almost all the F2s had a ratio of 3-to-1, where the 3 was whatever the F1 phenotype was, and then the 1 was always the parental trait that wasn't in the F1s. So 5,474 individuals had round seeds, 1,850 had wrinkled seeds. So it's really remarkable how many crosses that he actually did. Like, he was a firm believer in large numbers and a firm believer in doing these experiments right. And so he did this for all 7 of these characters again, and he found these rules were very consistent. So the F1 individuals all shared one of the parental, and then the F2s all had this 3-to-1 ratio or close to 3-to-1 ratio. So how is this possible? So how did Mendel explain these results? And so he explained these results is that each character was controlled by a single gene, and each one of these genes had 2 alleles. Each parent was homozygous for one of the 2 alleles, and the gametes produced by the parents contained one of the 2 alleles. And then finally, one allele was dominant over the other. So how does this make sense? So let's talk first about the tall dwarf. So we're talking about this particular character right here. And he said each character was controlled by a single gene with 2 alleles. So all individuals carry 2 alleles. So in the first case, because we said that they bred true and they're homozygous, these genotypes, there's 2 alleles that we specify, but they're both the same. So this individual right here that's tall carries 1 big D and 1 big D. This individual that's dwarf contains little d and little d. So now we go back to our chapter 2, and this is just, you can think about this as in terms of like the homologous chromosomes. So the individuals have a copy from the father, copy of chromosome 1 from the father, and a copy of chromosome 1 from the mother. And so in this case, these individuals have the same allele from both the father and the mother. This individual has the same allele from the father and the mother. So these individuals cross together, and so these individuals undergo meiosis in order to produce haploids, and the only possible gamete that this individual can produce is a big D. So the only allele it can pass on is the big D. The only gamete that this individual can produce is the little d. And so we know with certainty that if we do a cross between these 2 individuals, this parent is going to supply this, and this parent is going to supply this. And so as a result, the F1s are all going to be identical to each other, and they're all going to be heterozygote. So they're no longer homozygotes, but now they're heterozygotes. So they contain 1 big D and 1 little d. So all of these F1s have the exact same genotype. However, these F1s, even though their genotype is different, they share the phenotype with one of the parents. And this is where the dominance comes in. So the dominance comes in because for these crosses, individuals that are heterozygote have the exact same trait as the individuals that are homozygous. So these individuals are tall, just like these individuals are tall. So all of the F1s, all are tall individuals. So now in the next generation, he decided to cross the F1s together. And so now we get into the realm of probabilities. So why is that? So these individuals now can produce more than one type of gamete. So this individual is heterozygote. So this individual could either pass on a big D or a little d. And so this is just, you can go back to the rules of chapter 2, where we talked about the separation of the homologous chromosomes during meiosis 1. And so you could either form a gamete that is mostly your mother, or you could form a gamete that is mostly your father. And so because of that, both gametes are possible. And this was 1/2 and 1/2. So there was a 50% probability that this individual would make this, and there was a 50% probability that this individual would produce this. And so this got into his rule right here. So gametes are created by random segregation. So this was another observation that Mendel made, and that basically is what allows us to say if we have a heterozygous individual, it is a 50-50. It's a random combination of which gametes that get produced. So now we allow these gametes to form, and so there's 4 different possibilities of what can occur. So in one case, you have a big D from the father combined with a big D from the mother, and then the individual is big D, big D. In another case, the big D from the father combines with a little d from the mother. So these are the fathers, just to follow this a little bit better. These are the mothers. And you have a heterozygote individual. Or the father could produce a little d, the mother could produce a big D, and you have a heterozygote, and then little d's from both, and then that's the only way you can get a dwarf plant. So this is where we have the ratio. So if we look at the ratio of genotypes, the ratio is 1 to 2 to 1. So a quarter of the offspring are big D, half the offspring are heterozygote, and a quarter of the offspring are little d. But if we look at the phenotypes, the ratio is 3 to 1, and that's because we group these genotypes together because they have the same phenotype. And again, that's just the dominance relationship. So we started out with kind of terminology for the phenotypes, and now we have similar terminology for the genetic information. So the gene is the unit of inheritance. So this to me is analogous to the character. So a gene is also a sequence of DNA that codes for a molecule. So it can encode for a protein, encode for an RNA, but there's some sort of fundamental unit of inheritance. Genes can have multiple flavors, just like characters can have multiple traits. And those are called alleles. So the different versions of the alleles of the genes are called alleles. So it's one possible variant of a given gene. So for Mendel, there was only 2 alleles, but now we know that that genes could have more than 2 different versions. So what he studied was just a very limited case. So a genotype is the composite of an organism's alleles that influence the phenotype. So an example of a genotype is this. So you can have a genotype that has 2 genes, just like a phenotype can have multiple characters. And so we have to specify, so for these 2 genes, we have to specify what are the 2 alleles. So big A, little a, big B, big B. So this individual, we specify the genotype at gene A and gene B. A gamete is a haploid cell produced by an individual to produce a new individual. And each haploid cell has its own genotype. So this individual right here, could form this gamete right here, for example. So you notice the haploid only has 1 allele of each gene, and that's because haploid versus diploid. And then finally, genetic analysis is the process of determining the genotype-phenotype map by analyzing phenotypes in multiple generations of progeny. All right, so we started out with this cross, and this cross started out with individuals that bred true. And so we knew ahead of time that this individual must be homozygous. So what if we had a situation where we wanted to know the genotype of an individual, but we didn't know whether or not this individual bred true? So an example, let's say we have a tall individual. So we have an individual that has the tall plant. And if I gave you that individual and I said, what is the genotype of this individual? Well, there's some ambiguity because we know that the heterozygote and the homozygote both would be tall individuals. So is this individual big D or big D little d? So a Testcross is one approach in order to answer that question. So the way you do it is you take this individual and you cross it to a dwarf individual. So this individual we know is homozygous recessive because it's dwarf. And so if we cross this individual, we can now analyze the F1 to determine the genotype of this individual. And so we'll just write out the possible crosses in the 2 situations. So in situation 1, this is what would happen if the individual was this. In the Testcross 2, this is what would happen if the individual was this. So the homozygous tall would only produce the big D gamete because it's homozygote, and the homozygous dwarf would only produce the little d. So every single offspring from this cross would be tall individuals. In this case, if the individual was heterozygous, then this could produce 50% of each different gamete. So 50% of the gametes produced from this individual would be big D, and 50% would be little d. For the homozygous dwarf, 100% would be little d. And so if we looked at the offspring, we would find 50% would have this genotype, and 50% would have this genotype. And if we looked at the phenotypes, we would see half of them were tall and half of them were dwarf. So the Testcross allows us to determine the genotype of an individual by looking at the offspring. So the important things to remember is that you always cross it with a homozygous recessive individual, and then you analyze the F1 offspring that are produced.